Hello everyone, how are you all doing? This video is the third video of revising all the International AQA A level biology from PMT. In the last two videos, we've covered the biological molecules and we've covered them in two videos. In this video, we will be trying our best to finish cells. If not, it will be written in the title. In the cells of the AQA, we will be using the summary notes over here. I believe this video must be useful for the International AQA, Igcse, and I guess OCR as well. So let's get started with the cells. First of all, there are types of cells, but how do we know there are types of cells? We studied them, right? So there are many methods for studying the cells. And one way in which this is done is through the use of the microscopes, of which there are two main types, the light microscope and the electron microscope. The light microscopes use a pair of convex glass lenses that can resolve images that are 0.2 UM apart. The reason for this is that this is wavelength of light and therefore restricts the resolution that a light microscope resolves to. This is compared to electron microscopes, which can distinguish between items 0.1 nanometer apart. The magnification of an image as seen through a microscope can be calculated using the following equation. Magnification equals size of image over size of real object, which we say I equals I equals A over M. So M is equal to, no, wait. I equals M. We have this, right? So we have, here, I is equal M. So I am, okay? You can remember it this way. You need to know that resolution is defined as the minimum distance apart that two objects can be distinguished as separate objects in an image. The greater the resolution, the more clear the image will be. Now we go to the one after, which is the electron microscope, and it has some types as well, transmission and scanning, for example. The electron microscopes has limitation. The light microscope has limitation that it only resolves to resolution of 0.2 UM. Means the electron microscope can be used to look at objects that are closer than the 0.2 UM apart. That's why there are some components in the cells that can be seen using EM but never using the LM. So there are two main types of the EM. EM is basically electron microscope. These are the transmission electron microscope, the TEM, and the scanning electron microscope, which is the SEM. The electron microscope work in a similar way to the light microscope, but instead use a beam of electrons that are focused by electromagnets inside a vacuum environment. Highlight vacuum environment. The vacuum environment is needed so that particles in the air do not deflect the electrons out of the beam alignment. We highlight again the vacuum environment because it's gonna have a negative thing, a con. The following details how each type of electron microscope works. So, transmission electron microscope. A beam of electron passes through a thin section of a specimen. Areas that absorb the electrons appear darker on the electron micrograph that is produced. Scanning electron microscope. In a scanning electron microscope, a beam of electrons passes across the surface and scatter. The pattern of scattering builds up to a 3D image depending on the contours of the specimen. There are some limitations though when using electron microscope. The limitations for SEM and TEM are that the whole system must be in vacuum. I told you to highlight it. So living specimens cannot be observed. Also, a complex staining process is required which may introduce artifacts into the image. 3. Specimens have to be very thin, particularly for TEM, so that the electrons can pass through. 4. SEM has a lower resolving power than TEM, but both have greater resolving power than a light microscope. This is the comparison between LM and EM. Now, let's go into the cell fractionation and the ultracentrifugation. You know that the cell fractionation is the process in which different parts and organelles of a cell is separated so that they can be studied in detail. The most common method of cell fractionation is differential centrifugation. The process of homogenization is detailed below. First of all, the cells are first blended in a homogenizer, forming the resultant fluid called the homogenate. This tube of homogenate is then placed in a centrifuge and spun at a slow speed. The heaviest organelles, which is for example the nuclei, are forced to the bottom of the tube where a thin sediment or pellet forms. The fluid at the top, called the supernatant, is removed, which leaves just the sediment of the nuclei. The supernatant is then transferred to another tube and spun at a slightly faster speed. This time, the pellet that forms contains the next heaviest organelle, the mitochondria. This process continues so that each time the speed is increased, the next heaviest organelle is sedimented and separated out. The homogenate at the beginning is placed in a cold buffer solutions of the same water potential as the cells. This is to prevent the organelles from bursting under osmotic pressure, to inactivate any enzymes from breaking down organelles, and so that the pH does not fluctuate, as you see over here. Okay, so after is the cell structure. I want you to know that all living organisms are made of cells, of which there are several different types of cells. Some of them share some common features. Humans are made of eukaryotic cells, and all eukaryotic cells contain a nucleus and membrane-bound organelles. A more detailed structure of cells, called the ultrastructure, can be obtained by using a microscope. Here is the ultrastructure of eukaryotic cell, like the cell in your body. So it has a nucleus, which is a double membrane called the envelope. It contains around 3,000 nuclear pores that enables molecules to enter and leave. It also contains chromatin and a nucleolus, which is the site of ribosome production. A granular jelly-like material called nucleoplasm makes up the bulk of the nucleus. Second point, the rough endoplasmic reticulum is a series of flattened sacs enclosed by a membrane with ribosomes on the surface. The RER folds and processes proteins made on the ribosomes. Third, the SER is a system of membrane-bound sacs and produces and processes the lipids. Also, the Golgi apparatus is a series of fluid-filled, flattened and curved sacs with vesicles surrounding the edges. Golgi apparatus processes and packages proteins and lipids. It also produces lysosomes. Mitochondria. Mitochondria are oval shaped, bound by a double membrane called the envelope. The inner membrane is folded to form projections called cristae, with a matrix on the inside containing all the enzymes needed for respiration. The centrioles. The centrioles are hollow cylinders containing a ring of microtubules arranged at right angles to each other. Centrioles are involved in producing spindle fibers for cell division. Ribosomes are composed of two subunits and are the site of protein production. Lysosomes are vesicles containing digestive enzymes found by a single membrane digestive enzymes bound by a single membrane. Now we go to the prokaryotic cells, which are found, for example, in the bacteria. They have cell wall, rigid outer covering made of peptidoglycan, capsule, protective slimy layer which helps the cell to retain moisture and adhere to surfaces, plasmid, circular piece of DNA, flagellum, a tail-like structure which rotates to move the cell, pili, hair-like structure which attach to other bacterial cells, ribosome, site of protein production, mesosome, enfolding of the inner membrane which contain enzymes required for respiration. Viruses are non-living structure which consists of nucleic acid, either DNA or RNA, and is enclosed a protective protein coat called the capsid, sometimes covered with a lipid layer called the envelope. The cells of multicellular organisms are organized into tissues into organs, and organs into systems. Cell division by mitosis. The role of mitosis in the cell cycle is to produce the identical daughter cells for growth and asexual reproduction. All the cells produced by mitosis are genetically identical. Therefore, mitosis does not give rise to genetic variation. These are three stages of the cell cycle: mitosis, interphase, and cytokinesis. Mitosis is a form of cell division that produces identical cells. There are four stages of the mitosis: the prophase, the metaphase, the anaphase, and the telophase. The interphase, during the stage of the cell that grows and then prepares to divide. Chromosomes in some organelles are replicated. Chromosomes also begin to condense. Cytokinesis, during cytokinesis and the parent and replicated organelles have moved to opposite sides of the cell, and the cytoplasm divides, thus producing two daughter cells. The mitosis is very important as it's there in growth, repair, and reproduction. Growth, all the cell produced are identical, so organisms can grow using mitosis. Repair, all cell produced are identical, so organisms can replace dead tissues using mitosis. Reproduction, some single-celled organisms like yeast reproduce by dividing into two identical daughter cells by mitosis. The process of mitosis is summarized in the diagram below. So we have the G2, which basically loosely coiled replicated chromosomes. We have a centrome, we have a sister chromatid, and breaking down of nuclear membrane. And then the prophase and breakdown of nuclear membranes. When the fiber appear, chromosomes condense. Then in the Pro-metaphase, the spindle fiber attached to chromosomes and the chromosome condense. In the metaphase, the chromosome align. In the anaphase, the centromeres divide and the sister chromatids move to opposite poles. In the telophase, the nuclear membrane new forms and the chromosomes decondense, as well as that the spindle fiber disappear. For the cytokinesis, the cytoplasm divides and the parent cell becomes two daughter cells with identical genetic information. If you do not know about the binary fission, it's a process by which prokaryotic cells divide and occurs as follows. The circular DNA in the cells replicates and both copies attach to the cell membrane. Plasmids also replicate. The cell membrane then began to grow between the two DNA molecules and began to pinch inwards, dividing the cytoplasm in two. A new cell wall forms between the two DNA molecules, dividing the original cell and the identical daughter cell. Each have a single copy of the circular DNA and a variable number of copies of the plasmids. As viruses are non-living, they do not undergo cell division. Following the injection of their nucleic acid into other cells, the infected host cell replicates the virus particle. That's completely great. Now we go to the biological membranes, where I want to tell you that all cells and organelles are surrounded by a partially permeable membrane composed of C-phospholipids with protein molecules between the phospholipid molecule. The main function of the membrane is controlling the movement of substances in and out of the cell organelle. However, it also contains receptors for other molecules such as hormones and enables adjacent cells to stick together, so the fluidity of the membrane and the mosaic arrangement of the protein give the structure of the membrane its name, the fluid mosaic model. The cell membrane is comprised of phospholipids in which the hydrophilic heads point outwards and the hydrophobic tails point inwards. The structure allowed lipid-soluble molecules to pass through the membrane, but not water-soluble molecules. It also means that the membrane is flexible and self-sealing. The membrane contains many other components. These are detailed as below. Proteins, cholesterol, glycolipids, and glycoproteins. The proteins, these may be integrated throughout the membrane, intrinsic proteins, or maybe on the surface, extrinsic proteins. Intrinsic proteins include carrier proteins, which allow substances to cross the membrane. Proteins are present to aid movement across the membrane, provide mechanical support, and act in conjunction with glycolipids as receptors. The glycoproteins are carbohydrates that attach to extrinsic protein and act as self-respicating membrane. And nitrous transmitters. These allow cells to recognize one another as well as attach to form tissues. Glycolipids are made of carbohydrate with lipids. These extend from the surface of the cell and act as a cell surface receptor for certain molecules. They also allow cells to adhere to one another to form tissues. Lastly, the cholesterol. And it's present to make the membrane more rigid and reduce the lateral movement of the phospholipid. It also prevents the leakage of water and dissolved ions from the cell, as it's very hydrophobic. This is the end of this video, and inshallah the next video will be finishing the cells. I wish you find this video very useful. Have a nice, beautiful day.